Hi, welcome to the applications of nanoparticles, or to be specific, a deep dive about the applications of magnetic nanoparticles. I am Michael Zircon, and let's go! The size of nanoparticles gives them the advantage of going into the body and can be divided into a small scale to cure diseases. Due to its advantage of its size and shape, magnetic nanoparticles have a great advantage in the usage in biomedicine. It can help in cell separation or help in drug delivery for curing diseases such as hypothermia and cancer. In biomedicine, it is often necessary to separate blood and biological entities from an environment in order to analyze blood sample or body tissue. For example, we need to localize a certain blood tissue or blood sample in order to eliminate the possibility of contamination. For example, if only we need is blood samples that are red blood cells, we need to eliminate the white blood cells so that it cannot interfere with our readings. That's an example.
One method of sperm selection is called MAX. This technique uses magnifying particles to bind low-quality sperm to each other. The human body has a system for the disposal of inappropriate cells. MAX takes advantage of this system in the selection of quality sperm. To remove poor quality sperm, magnetic particles are used, which bind to a special marker at the surface of the sperm. With the action of a powerful magnet, only healthy sperm with the best chance of fertilization are left in the sample and used for fertilization.
But how does nanomagnetic particles work? Let's take a look. The supernatant, or the unwanted particles in the solution, are passing through the magnet at the beginning, while the supernatant is absorbed by the magnet or it is adsorbed. For example, it is attracted to the magnet. The desired solution can pass through without any supernatant in between. So the magnet will catch all those unwanted particles so that the one that will pass on our passageway would only be the solution that we want to use. Okay, here is an example. We have a solution of targeted cells and non-targeted cells. The non-targeted cells are actually the cells that are beneficials or the cells that we want to have in a solution. And the targeted cells are the ones that we should eliminate. Now in these mixtures, how can we separate the non-targeted and targeted cells? So with the use of nanomagnetic particles, the nanomagnetic particles will target the targeted cells. They will use their magnetic properties to attract the targeted cells towards their polar force or polar area. Then it will be stuck, leaving freely our non-targeted cells to pass through and be used. The magnetic field aboard the supernatant, or the unwanted, to one side while the fluid can go through the area without the supernatant involvement inside the solution. It can separate unwanted biomaterials from the magnetically tagged, which is gathering around the magnet due to the magnetic property. As shown in the video earlier, it is used by the magnetic field to attract the targeted cells so that they cannot mix on our non-targeted cells. With this, it can be used in many parts of biomedical applications, such as separating the rare tumor cells from the healthy cells. As we can see, it is a problem actually on segregating the healthy cells to those who are unhealthy ones. With the use of our medicines today, we often target both healthy and non-healthy cells. So instead of it being beneficial, it actually damages us too. With this, we can use it to target low-number cells, leading to enhanced detection of materials in blood and process technology for polymerase chain reaction. Also, for the separation applications, it can be used in optical sensing due to high sensitivity of magnetic nanoparticles. If the target material is a solid matrix, it can act as the surface to locate the target materials and to increase the concentration.
Presense sensors work on the principle of fluorescence quenching and allow O2, CO2, and pH measurement. The sensor is excited with light from a LED and emits fluorescence in response. Depending on the presence and amount of analyte molecules, this fluorescence response varies and can be translated into the respective concentration. Presence oxygen sensors also allow determining oxygen content in the gas phase with the same precision as in the liquid phase. Non-invasive measurement.
Now, let's talk about how nanomagnetic particles can be used on drug delivery. The magnetic nanoparticles have the advantage of treating the area, or the target area more specifically. Due to the guidance of a magnet, it can be delivered into a specific site, then release the drug. Since the drug is used only in a specific site, it can reduce the dose amount, meaning we can have a less overdosing and less side effects towards the body because we only use small amount that is beneficial or what is needed to that certain area.
Cancer cells isolated from human tumours can be used to help the development of novel cancer therapies. For most drugs to be therapeutically active, they must go inside the cell. Nanoparticles carrying drug payloads can be used to facilitate the drug delivery. Nanoparticles can be targeted to receptors that are overexpressed on the surface of cancer cells. They then enter the cells via a process called endocytosis. Antibody fragments engineered on the nanoparticle bind to the receptors, initiating the internalization process. Inside the cell, clathrin proteins form a cage around the developing endocytic vesicle, which is then cleaved from the cell membrane. The newly formed vesicle carrying the nanoparticle travels further inside the cell, where it is captured and fuses with an early endosome. Inside the acidic endosomal compartment, the nanoparticle degrades, releasing the therapeutic cargo, which induces a controlled death of the cancer cell. Nanoparticle-based drug delivery provides a novel and targeted approach for the future of cancer therapy.
So how are these nanoparticles going to work? We're going to take our nanoparticle and fill it with our cancer drug right there. And then we're going to attach a targeting ligand to the surface of the nanoparticle. And I'll explain how these work in just a bit. Once we do this, the nanoparticle is injected right into the bloodstream. Here's what it looks like. Above and below the bloodstream on the left side are all the healthy cells. These are the good guys, which we want to keep alive. The nanoparticle can't attack them because of this tight junction blood vessel, which prevents our nanoparticle from reaching them. On the right hand side here are all the cancer cells, which we want to attack. And how do we do this? Well, all these cancer cells have a receptor ligand on their surface and are surrounded by leaky blood vessels. This is good because it allows our nanoparticle to have access to these cancer cells as it makes its way through the bloodstream. So, as the nanoparticle nears these cancer cells, the targeting ligand on its surface can bind to the receptor ligand on the surface of the cancer cell and cross the leaky barrier to eventually kill it. This is the big thing that separates treatment with nanoparticles from chemotherapy and radiation therapy. Because of all this, we're going to reduce the amount of healthy cells being attacked as well as increase the amount of cancer cells that die. And all of this is going to make for a happier, healthier you.
It can be seen here that the drug is first coated with a magnetic material and then injected into the body. The external magnet guides the nanoparticles into one specific site along the blood vessels. There's actually a magnet that controls the outside movement of the nanoparticles. Then, when it arrives at the target site, it can be attracted at a tumor tissue, which releases the drugs due to the change in internal environment, such as a change in pH, either high pH or low pH, either due to acidity or basicity of the tumor. In the previous study that is done by Pankhurst, Connolly, Jones, and Dobson on 2003 on rabbits, there's actually no side effects of toxicity of the drug delivered. They give it a 30-day trial or a 1 month, and the result is actually encouraging. There's no effect. So the magnetic nanoparticles can be used in a kind of drug delivery. However, human trials are not yet done. So we're hoping for the best of these magnetic nanoparticles. So, that is our deep dive about our magnetic nanoparticles. We can use it on cell separation or in drug delivery. So it's actually a promising future for us. So thank you for listening and have a blessed day.